Hello everyone, thank you for watching Dennis Insider. Today we're going to talk about immunosuppressive drugs, and in the outline we will first talk about why we use immunosuppressive drugs, then we will talk about the normal immune response, and then we will talk about all of the targets or the mechanism of action for these immunosuppressive drugs. So first, why use immunosuppressive drugs? They're mainly used to prevent transplant rejection, treatment for autoimmune disorders, acute or chronic allergy, cancer. But the thing with the immunocompromised patient is that they're more prone to infection and cancer, but also if someone is on immunocompromised drug, their side effects is being more prone to infection or cancer, lymphoma, and such. Next, we talk about just the normal and immune response, and this will help us figure out what are the targets that are, what are the targets or mechanism of action for these drugs. So first here we have immune response. We have a antigen that comes in. So first step is we are sensing the antigen, and you can see here that's sensing the antigen. Then we have the activation, which is the signaling downstream to activate via transcription. So here you have immune activation via transcription, and it will enter the nucleus. Translocation into the nucleus here will have more downstream signals that will produce cytokines. And finally, the cytokines will activate or activate T cell proliferation. And here, this is our final step. So step one is immune sensing. It will cause a downstream signal, immune activation, transcription in the nucleus, which will release cytokines, and the cytokines activate T cell proliferation. So these, it's right here where I circled is where our targets will be for the drugs. And just as the overview first, and then we'll jump into the detail. Here we just have some list of drugs that we will talk about. So here we have the mechanism of action. The first one is the decrease of inflammatory cytokines. And as we mentioned, after the nucleus will cause the cytokine production, so we can inhibit the steps right here by using glucocorticoids and TNF alpha antibodies. Glucocorticoids inhibit cytokines and mostly used for acute infections, acute illnesses. TNF antibodies cause non-functional cytokines, so you still have cytokines, but they will not cause T cell proliferation, and that is used for chronic use. Next, we have inhibition of T cell activation, and I'll just over and over repeat this as we go on. There's two. We have inhibition of T cell, and then in C, we have T cell depletion. So in B, we have normal T cells, but they're non-functional, and in C, we have totally no T cells. So first, in inhibition of T cell activation, we have calcineurin inhibitors, and they block the activation. So after immune sensing, we have the immune activation, and so if we inhibit the immune activation, we don't get transcription, and then we don't get cytokines or T cells. Number two, we have Ctla4 Mimetics, and these block the step one, which is sensing up top. So if you block sensing, basically there's no immune response at all. Then we have inhibition of IL-2 receptor, and then fourth is inhibition of IL-2 receptor signaling. So the first inhibition of IL-2 receptor is right here where the cytokines will not activate T cell proliferation, but inhibition of IL-2 signaling, it inhibits the downstream signal that you do not get T cell proliferation. So they're similar but different drugs. And then third, or C, is T cell depletion, where we don't have any T cells. We have two types of mechanism. We have Cd3 antibodies that end in either NOB, which are non-humanized antibodies, and then MAB. And then we have anti-thymocyte globulin, which blocks T cells from every different angle on the T cells, not just one single receptor like the Cd3 antibody. Lastly, we inhibit cell proliferation, and these are antimetabolites. So this is back to basic biology, where they will inhibit either the S phase or inhibit mitosis. So that's the overview. We'll be talking about these four mechanism of actions. decreasing the cytokines, inhibiting T cell activation, total T cell depletion, or inhibiting cell proliferation. Now let's jump into it. So once again, the first mechanism of action we're talking about is decreasing of the cytokine, meaning there will not be an immune response. So that's what we're doing here. No cytokines, meaning there will not be any T cell proliferation. So first we have is at first, we will talk about all the mechanisms. So like we said, the first one is decreasing the cytokines. Second is inhibition of the T cell activation. Third is total T cell depletion. And fourth is inhibition of the cell proliferation. So in the first one, where we're reducing the cytokine production, which is right here. Oh my God. So first we have the cytokine production, and that will cause no immune response. So we have two examples here. We have the glucocorticoids, and then we have the TNF alpha antibodies. So glucocorticoids such as prednisone and hydrocortisone, they're used once again to prevent transplant rejection, suppress allergy, inflammation, and autoimmune, just like all the other immunosuppressive drugs. In a normal immune response, we get a cytokine production, which will activate T cell proliferation, but here you will not get that. So when you give a glucocorticoid treatment, what happens is that normally your adrenal glands secrete endogenous glucocorticoids during your sympathetic parasympathetic response to suppress the immune system. Now, when we give the steroid or glucocorticoid, it crosses the membrane to displace a heat shock protein called HSP90, which binds, the glucocorticoid binds to the glucocorticoid receptor and is translocated into the nucleus. So once we have the glucocorticoid on the glucocorticoid receptor on the DNA, that will change the gene expression. Changing the gene expression in the nucleus will basically inhibit immune response, and we do not get any cytokines. So that's what we will show right here. So here we have the glucocorticoids. Here we have the glucocorticoids in yellow. We have the inactive glucocorticoid receptor right here. Then we have the glucocorticoid, the heat shock protein is basically displaced, and now we have the glucocorticoid plus the glucocorticoid receptor that will enter the nucleus. In the nucleus, it will cause a gene expression, and we do not get this response to make cytokines. So it stops right here. So what is the mechanism of action for glucocorticoids? They're for acute use only, where we'll see the second category, which is TNF antibodies, are for chronic use. So they inhibit NF-kappa B gene expression, they inhibit the immune mediator and gene targeting for acute treatment. Next, we have just the adverse drug reaction or side effects to these. So the side effects are basically due to the increase in endogenous glucocorticoid. So with chronic use, we get something called a Cushing's syndrome. Symptoms such as hypertension, infection, type 2 diabetes, psychosis, weight gain, osteoporosis. What happens is your normal glucocorticoid levels increase glucose, and it's actually meant to help you in the flight or fight response. But here, what happens is that they increase your glucose production, they inhibit osteoblast function, and they promote bone resorption, causing us to basically get the Cushing syndrome. So that was the first one, glucocorticoids. We have such as prednisone and hydrocortisone. Like we said, their function is to basically inhibit all cytokine production by going into the nucleus and changing the gene expression. And it's important to understand that you should know these mechanisms of actions more importantly than memorizing drug names. Next, in the inhibition or decreasing in the cytokine category, we have the TNF alpha antibodies, such as Humira and Enbrel. here we have is the TNF antibodies. TNF alpha cytokine promotes an immune response. So now how do we fight that? So we have the drugs that will, the antibodies bind to the cytokines and they prevent the immune response basically. So in this stage, we do have cytokines, basically NF kappa B, all of that worked, and we got all the way to this step where we do have some cytokines. But now in green, you can see the antibodies, they're going to bind to the cytokines, and that way we do not get that downstream signaling for the T cell activation. So they basically bind and not let the cytokines be functional. The important part here is that the treatment, why you would use these TNF antibodies, is more for chronic use, such as arthritis, psoriasis, Crohn's disease, colitis. And just like every immunosuppressive drug, we have increased risk of infection and increased lymphoma. So once again, in the previous example, we repressed the expression of cytokines, so we had 0 cytokines. Here we have cytokines, but after binding to the TNF alpha antibody, they cannot produce immune response. So that is category 1, which is decreased cytokine, either no cytokine or non-functional cytokine. Next we have these are next two categories that we're going to talk about. We have the inhibition of the T cell activation, and then the third is the T cell depletion. So this is similar to what we just talked about with cytokine. In number 2, we have normal T cells, but they're non-functional. But in the third, we have no T cells. So just like before, either we had no cytokines or non-functional cytokines. Here we have either no T cells or non-functional T cells. So the first category in inhibition of, and then just going back to our image, once again, here we are attacking our T cell proliferation step here, right there, after the cytokine. So what happens is that here is the normal reaction that takes place, or normal immune response. We have a antigen that comes in, antigen presenting cells will present that to the T cells. We get the immune sensing. We have 2 signal basically dependent here. We get the immune activation after we get that. This is important, is that this is a calcium dependent immune activation. So we get the calcium influx that comes in first. The calcium will bind to calmodulin, and that forms calcineurin. And this will be our mechanism of action. This is a target that we will talk about in inhibition of T cell activation. Calcineurin binds in, it dephosphorylates an NFAT compound. The NFAT can then go into the nucleus to transcribe genes like the IL-2, and then you get the response. So NFAT goes into the nucleus, you get some gene transcription, and now we make the IL-2 cytokines. So what happens in the calcineurin inhibitor is that we know this is a calcium-dependent immune response. The drugs are called cyclosporine or tacrolimus. So their mechanism of action is to inhibit that calcineurin that we see right here. Once we have calcineurin, basically we will not have, we cannot activate the proteins, we cannot dephosphorylate NFAT. NFAT does not go into the nucleus and make IL-2, so it stops right there. Treatment is for either oral or topical use. Once again, like every immunosuppressive drug, we get that. But the more important, we have nephrotoxicity here, and we have some neurotoxicity here. Because this mechanism will disrupt all of the calcium-dependent pathways, and we have a lot in our normal homeostasis, such as muscle contraction or metabolism and such, that are calcium-dependent. So this is easy to remember: calcineurin, calcium. Next we have Ctla4 Mimetics, and this is Cept. So first we have the sensing, which is, as we know from immune response, needs a 2-signal requirement to get activated. So we have the MHC2 right here, and we have the T cell receptor right here on the T cell. Then we have the second signal, CD80, on the antigen presenting cell CD80 or CD and CD86, and then we have CD28 on the T cell, and this is the target that we're going to focus on here. So what happens is that here we get inhibition of T cell activation using Ctla4. So here we have something new, Ctla4. What happens is that Ctla4 is also competing for CD80 and 86 that normally should bind to CD28. But when we have too much T cell activation, a big response, the body basically will try to repress it or turn off the signal, and we use the Ctla4 to turn it off. So just important that if we have more CD28 than Ctla4 that can bind to the CD80, we get T cell activation. But if we have more Ctla4, then we get T cell inactivation. Hope that made sense. So the drug that we talked about, that we have talked about for the Ctla4 mimetic is called Cept. And with Cept, the mechanism of action is that it's binding CD80 on the antigen-presenting cell and blocks the CD28. So there is no T cell activation. So here is the Cept. It will basically compete and bind CD80, as we can see here. So therefore, CD80 can neither bind to CD28, and it kind of binds to a mimetic or a fake copy of Ctla4. So the treatment is that it's given IV to prevent mostly organ rejection, kidney. You can limit the nephrotoxicity with calcineurin, and once again, this is more for chronic use. So we're talking about the inhibition of T cell activation. The first one we just talked about is the Calcineurin Inhibitors, and then we talked about the Ctla4 Mimetics, and now we talk about the inhibition of IL-2. So we have IL-2 inhibitor, but then the next one we'll talk about is IL-2 receptor inhibitor, and the difference here is that one ends in MAB and one ends in MUS. So here we have in IL-2, once again, it's a normal response. We should have a 2-signal requirement. We get the calcium dependent, dephosphorylate the end fat, and goes translocates, nucleus, makes some cytokines. And then we have the IL-2, which activates the IL-2 receptor using a mTOR pathway, and that will cause T cell proliferation. So here we are targeting, like we said, it's an IL-2 inhibitor. So here we have the drugs, MAB, and what they do is basically they come in here in the orange and they block the receptor. So here we are blocking the IL-2 receptor, MAB bind to the IL-2 receptor and block the T cell proliferation. Therefore, there's no mTOR pathway and such. So the treatment is the first drug is mostly for kidney transplant, used with cyclosporine and glucocorticoids. And then we have the second drug, which is no longer used due to liver toxicity. So basically, in this one, we are inhibiting T cell receptor, and in the next one, we are inhibiting the motor MTOP inhibition pathway. So the receptor is still good, it gets activated, but the downstream pathway to cause T cell activation is blocked, basically right here. This is blocked for this. Okay, so mTOR inhibition is important because this is this helps the cell grow based on the nutrition. So if the cell has all the proper nutrition, basically all the food in the pantry ready to go, it will tell the cell that you can grow. If it does not have the proper nutrition, then it will not allow that to happen. So this is basically a regulator for cell growth. And the mechanism of action is that if we inhibit the mTOR pathway, we block the downstream signaling of the cytokine receptor, and then we saw that there will not be T cells. So the drugs here, usually for kidney transplant, coating of the stent to suppress the locally area where in the artery where you're putting the stent or anti-cancer use. Some drug reaction that we have here is that we have improper wound healing. Uh, Basically, you do not get any growth or proliferation of cells because of the nutrition, thrombocytopenia, hyperlipidemia. So basically, you have impaired wound healing and so on and so forth. Next, we talk about is our T cell depletion, where basically, so far we talked about how we can attack the cytokines, how we can attack the receptor, or how we can attack the motor pathway here, so we don't get T cell proliferation. Now, lastly, we just talk about how we have zero T cells. Number 3, we talked about total T cell depletion. We have two types here. We have the Cd3 antibodies and then anti-thymocyte globulins or thymoglobulin. So first we have the normal immune response mechanism. So here we have that CD3 are presented on all T cells. The MAB, monoclonal antibodies bind to the CD3. They activate CD3, and they will also bind a natural killer cells, causing ADCC, which stands for antibody-directed cytotoxicity. That causes a cytokine storm and basically destroys the T cell. So here we have the ADCC cytotoxicity happening, where we have this antibody, Cd3 antibody. The NOB stands for its non-humanized antibody. So what's happening here is that in this side is non-humanized, but the red side is humanized. So the body is able to attach to the CD3 on one side, but on the other side, which is foreign, the body does not recognize it, and then it will go and attack it, and that will basically kill the T cell. And this is in layman's term. So first we have the CD3 antibody, the NOB. Their mechanism of action is once again to produce cytokine and cause a ADCC, antibody-directed cytotoxicity. The side effect is that we get a cytokine release syndrome, or another term is cytokine storm. So we only use this for acute treatment. So that is the first generation, the NOBs. The second generation, which is MAB, these are more human-like. So here, what you will see that we lack the FC region. So there is no cytokine response, but causes an immunotolerance. So as you can see here, we don't have that non-humanized or the foreign region that we saw on the previous one. So the body here basically will not cause that ADCC when the natural killer cells will not come in and attack it because there's no foreign receptor. But what happens is that the cell basically can sense that there's a blocked receptor. They will express the TGF beta, increase T regulatory cells, which are good, and that will calm down the immune response, in other terms cause immunotolerance. So not lacking the FC region, there's no natural killer cell. No natural killer cell will cause basically the cell to say, what the heck is going on? Let's check this out. Increase TGF beta, increase the regulator, and then we get the basically immunotolerance. So side effects, once again, cytokine storm, and then it's basically should be used for chronic use for the second one. So that's important here. And then we treat for type 1 diabetes, multiple sclerosis, or autoimmune diseases. So this is more immunotolerant, whereas the previous one, first generation, no T cells at all. Then we have anti-thymocyte globulin. These are polyclonal antibodies. Thymoglobulin is the drug name. So these polyclonal antibodies to T cells promote T cell depletion. Thymoglobin is many different antibodies. So they come from every angle that they can attack. They're not monoclonal, where they can only attack one specific target. So here, basically, they can come in from the natural killer side, cause a cytokine storm, and come from different angles. So basically, mechanism of action is total T cell depletion using the polyclonal antibodies. So we use it for acute organ transplants. Drug reaction, again, leukopenia, the cytokine storm. Lastly, we talk about the inhibition of cell proliferation, mostly S phase or mitoses. So these drugs are called antimetabolites. Um, we have two types. We have one that can inhibit the DNA, so those are these methotrexate or purine analogs, and then some that promote DNA damage, the amides. So we know immune cells are the most rapidly dividing cells, and then cells require metabolites to divide. So we have two ways that we can block the cells, and we know this from biology. We have the S phase here, and this will be our first mechanism of action, or the target, where we block the building blocks for the DNA in the S phase, and then there will not be DNA. Or we can inhibit in the mitoses side. So you inhibit the microtubules. Basically, the daughter cells are not able to separate, daughter chromosomes are not able to separate, and that will inhibit the DNA. So inhibit the cell division, use either attacking the S phase or attacking the microtubules and mitoses. And then basically we don't have a new strand of DNA, and if DNA damage, it cannot be replicated correctly, so there's no new copy made. So we basically use this for autoimmune diseases like everything else, anti-cancer therapy, but the side effects here are infection, GI toxicity, fatigue, and alopecia. So that was immunosuppressive drugs. Once again, I will just drop a quick summary here again. We talked about first the decreasing of the inflammatory cytokines, and here we had the glucocorticoids, which basically inhibit or suppress the cytokines. You don't get any cytokine production, but in the second one we get the non-functional cytokines. Similarly, we can inhibit total T cells, or we can have total T cells, inhibition of T cell activation, which is similar, that it's a non-functional T cell, or we can have total T cell depletion. So in inhibition of T cell, we had the calcineurin, that's calcium dependent, blocks the activation. We have the Ctla4 Mimetics, that they basically they block the receptor, the second signal, CD80. It binds to CD80 and basically blocks sensing. Then we have either inhibition of IL-2 receptor, where we block the receptor and we don't get the downstream signal, or we block the mTOR pathway. The mTOR pathway here. Third, we have total no T cells here, so we can either have the Cd3 antibodies, the non-humanized or the humanized monoclonal antibody, or we have the polyclonal or thymoglobulin that will basically block T cell from every angle. Lastly, we can inhibit cell proliferation using the antimetabolites that either attack the building blocks in the S phase, or they attack the microtubules in mitoses. Thank you so much for watching, and sorry my allergies are just acting up.